Welcome to Tsat. Today I am going to discuss regarding cloning and stem cell technology. So, why we need to discuss cloning and stem cell technology together? Because cloning is possible with stem cells. So, to understand what cloning is, we need to understand what stem cells are. So, what are stem cells? Stem cells are certain cells present in our body which has got the capacity of replenishing and differentiation. Replenishing means creating their own copies through the process of cell division, and differentiation means one type of cell, the primary stem cells, when they are able to convert into different types of tissues and cells which have got a different structure in different organ system, is known as differentiation. For example, the cells which are formed from the zygote after the union of sperm and ova through the process of cell division forms into a structure known as morula. In the case of morula, which is made up of different cells which are the resultant of cell division from zygote, are anatomically similar and identical. These similar cells in morula differentiate and form into different tissues in different organ systems. For example, some of the cells in morula are responsible for the formation of brain. Some of the cells in morula are responsible for formation of heart. Some of the cells in morula are responsible for the formation of kidneys. For example, the cells in the brain are neurons, structurally different from cardiac tissue in the heart and different from structurally the nephrons in the kidneys. Their source is the cells in the morula. So, this capacity is known as differentiation. So, if the differentiation capability of a cell to create a complete organism is known as its totipotent capacities. So, certain primary stem cells, or otherwise also known as embryonic stem cells, have got totipotent capacity, where it means the capacity to create a whole complete organism is known as a totipotent capability. So, because of these differences in the capacities of replenishing and differentiation, the stem cells are being classified into different variant types. So, in this process, these stem cells, as I have mentioned, which have got these two capacities, the replenishing and differentiation. Replenishing is creating their own copies. Differentiation is converting into different cell structures to perform a specific function depending upon the organ system in which they have been formed. So, based upon replenishing and differentiation capability, the stem cells are being classified into embryonic stem cells, otherwise also known as primary stem cells, perinatal stem cells, and adult stem cells. Adult stem cells are also known as matured stem cells. So, we need to know where these stem cells are and their capacities. The use of human embryo stem cells raise an ethical concern, because the concern is, for an embryo which is developing in the mother's womb has got equal fundamental rights or any other rights a citizen has got. So, can we extract the stem cells, the primary stem cells which are available in the developing embryo, which may lead to disturbance of development, or the newborn child might be deprived of a certain organ because you have received certain amount of stem cells in the process of development. So, because of this reason, there are certain ethical concerns where you are not supposed to disturb the developing embryo. Then the question is, how to do research and development where innovation is not possible without research and development. So, the government, according to certain clauses, we have made certain conditions where you can do research and development with certain kind of embryonic stem cells which have been used in the process of in vitro fertilization. Experimentally in the fertility centers can be collected, which are not under use for the purpose of experimentation and research and development. There is no possibility of innovation and experimentation. So, that is the reason why there are concerns regarding collection of embryonic stem cells for the purpose of research and development, which may deprive the developing embryo. So, in this context, how do we differentiate these? Where do we have these cells and how they are different from one another in terms of their capacity of replenishing and differentiation? Embryonic stem cells are stem cells which are being extracted from the first couple of weeks after the fertilization at the stage of morula and gastrula stage of embryonic development. So, these cells have not much undergone differentiation. So, because these cells have not used their capacity of differentiation much, they will be having more capacity to replenish and differentiate. These are known as primary stem cells because they are the first cells which have been evolved from a zygote. These are also known as embryonic stem cells because these are the stem cells extracted from the embryo. When you relatively compare with perinatal stem cells and adult stem cells, the capacity of embryonic stem cells or primary stem cells replenishing and differentiation capability means the capacity to form new tissues, organs, organ systems and organism. When relatively compared with the rest of the stem cells, embryonic stem cells have got higher potency. That is the reason why these are known as a totipotent, means total means the capacity to generate a complete organism. Perinatal stem cells. So, here in the case of perinatal stem cells, so for example, these are primary stem cells. So, these are the stages. This is a stage of morula, this is the stage of morula, this is blastula. So, when we are trying to extract cells from the stage of morula and blastula in this process of development is known as, these are considered as primary stem cells. Primary stem cells can be used to develop these kind of organ systems like circulatory system or nervous system etc., which can be used for organ transplantation. This is possible with the help of primary stem cells. Pluripotent means which can be converted into many organ tissues. Totipotent means which has got the capacity to convert into a total organism itself. So, this is how at different stages primary stem cells or embryonic stem cells are being collected from the morula or blastocysts. The blastula is a stage after morula. So, the second variant of stem cells known as perinatal stem cells. These are stem cells which doesn't have much differentiation and replenishing capability when relatively compared with embryonic stem cells, but they are effective than adult stem cells in terms of replenishing and differentiation for organ synthesis or organ development. So, what are they, where are they, how to use them is a question to us. So, these are perinatal stem cells are being extracted from cord blood, cord tissue and amniotic fluid. So, this cord blood and cord tissue is can be extracted from the umbilical cord. Umbilical cord is a connection between the developing embryo and the mother to a tissue known as placenta. So, this is a cord which contains blood vessels, which supplies nutrition and oxygen to the developing embryo from the mothers. So, from the mother's blood there is a tissue in the uterus, temporary tissue in the uterus known as placenta. This umbilical cord is connected to the placenta and extracts the nutrition from the blood of the mother, supplying it to the developing embryo. The blood is not being supplied to the embryo, only the nutrition and oxygen is being supplied, because the blood group of the developing embryo might be different from the mother, which may lead to complications. So, this has been designed naturally in such a manner where the blood of the mother is not mixing with the blood of the baby. It supplies only essential nutrition and oxygen. So, this is a tissue which has got certain stem cells which can be extracted and preserved for about 100 years in cryogenic conditions. So, we have, for example, in our Hyderabad we have got stem cell banks. So, anyhow this is a tissue which is going to be wasted, which is discarded after the birth of the child. So, here because this is a tissue which can be processed and preserved in the stem cell technology, where this can be stored for 100 years, which have got the capacity of replenishing and differentiation, where they have got the capacity to generate organs which can be used for future transplantation if at all there is a necessity of an organ for our future treatment. So, in this context, to use this technology in future, we need to store this tissue in cryogenic conditions in stem cell banks. So, as far as stem cell banks are considered, we store these stem cells in cryogenic condition, and as you maintain an account in a bank, in the same way we need to maintain an account in stem cell banks where these stem cells are being stored. So, after as per requirement we can give order to the bank to synthesize the respective tissue for organ transplantation or plastic surgery. So, these are this is a cord, umbilical cord connected to the placenta, supplying the nutrition to the baby. So, which has the capacity to generate certain organs and organ systems. So, this is the graph which shows you different countries which are using cord blood and cord tissue for the purpose of therapy. Therapy means treatment for different kinds of diseases which an individual suffers in future. So, why we need to store these stem cells which are derived from the cord blood and cord tissue? Because the main reason behind it is, in the current scenario, in the process of organ transplantation, there is a problem in terms of organ rejection. Even though the blood group is the same, there is no perfect match of blood group to you. Even though the blood group is the same, there are certain proteins, there are certain antigen antibody reaction where to an extent rejects the transplanted organ. To avoid this transplantation rejection, what we need to do is the doctors prescribe certain medicines for a patient for lifetime to suppress the immunity to avoid rejection of the organ. In this process, the immunity rejects the foreign body. In this process, when the immunity is being suppressed, the patient is going to be impacted by different variant types of opportunistic infections, where the patient is going to suffer a lot with this kind of opportunistic infections. So, when organ through this process of stem cells are developed from the umbilical cord, the rejection does not happen because the organs are being developed from your own tissue which is being generated from you, and the tissue which is being stored in stem cell banks can be used for all the siblings. We need not store the umbilical cord blood and cord tissue of all the siblings. Storing one sibling's cord blood and cord tissue will be sufficient enough to treat all the siblings, not only the siblings, for both the husband and wife, not only to the husband and wife, to both the grandparents, maternal grandparents and paternal grandparents can be treated well. So, genetics treat both the maternal and paternal grandparents in the same way, even though we have certain cultural differences. Equally, stem cells derived from cord blood and cord tissue can be applied for siblings, husband and wife, maternal and paternal grandparents. So, these are the different relatives of a newborn child when the cord blood and cord tissue is being stored, can be used to treat these different individuals. That is one of the main advantage with this technology. And this is a technology which is not far away from us, which is not out of our reach. To use this technology, we need to store this tissue, and it is a proven technology. And in countries like UK and USA, there are certain organs being made like kidneys, or a part of spinal cord is being generated where a person has lost his spinal cord, a piece of spinal cord is lost in an accident leading to the lower limbs got impaired. So, part of spinal cord which is been generated through stem cell technology connected the lost connection, led to the limbs functioning again. So, this is not a technology which is far away, and this is a technology which has entered into our city in Hyderabad, where in 𝗹𝗶𝗻𝗁 Eye Hospital, a cornea is being generated with the help of stem cell technology, which is so this is a technology which is not out of our reach. It is under the process of development. Within a decade or a couple of decades, we are going to completely use this technology for making organs in the laboratory which can be used for transplantation without any resection. So, for this technology in the future to use, we have to store the stem cells. So, the possibility we have to store this kind of perinatal stem cells is storing our children's cord blood and cord tissue. So, the third variant of stem cells is adult stem cells, or otherwise also known as matured stem cells. So, what are these adult stem cells and matured stem cells? Let us examine. This is cord blood or cord tissue which is been taken from the umbilical cord, which in general will be discarded. So, this will be processed and tissue will be extracted and stored in cryogenic conditions in stem cell banks. Next is matured stem cells. These are stem cells which are present in our body. So, there are certain stem cells which are present in the bone marrow, inside the bone, or there are certain stem cells which are present in the subcutaneous layer of the skin, or there are certain stem cells which are in the inner lining of the intestines. So, these are the different sources of matured stem cells. But when relatively compared, as far as matured stem cells are considered, the differentiation and replenishing capacity of these matured stem cells in general is less relatively compared with perinatal stem cells and primary stem cells. But to an extent they can be used for the development of certain tissues in the case of plastic surgery, etc., or development of antibodies for the immunity to strengthen. So, this is a bone marrow. From the bone marrow tissue, different types of cells like RBC or WBC or platelets, different variant types of WBC like lymphocytes, etc. All these different variant types of blood cells are being synthesized from a similar tissue present in the bone marrow. So, this is an example of a synthetically prepared skin which is biocompatible from the stem cells. So, this is a initial stage of technological development of synthesizing skin in the laboratory, which is equivalent to a human skin with the help of stem cells which are natural, which will be accepted by our body, which can be easily be used in plastic surgery. So, these are the skin stem cells. These are the stem cells which are present beneath the epidermal skin, which can be used for stem cells in plastic surgery especially. These are perinatal stem cells. So, these are the cells just now I have discussed with you. Researchers have discovered stem cells in amniotic fluid as well as umbilical cord blood. These stem cells have the ability to change into specialized cells as we like. They can be synthesized into certain tissues in plastic surgery and organ tissue transplantation especially. So, because of disorders such as Parkinson's disease, severe burns and damages to the spinal cord can be rectified with the help of stem cells by generating different tissues and organs. So, what is happening here is there are certain diseases which are the resultant of degeneration of organs. To some diseases like Parkinson's diseases, we do not have certain perfect permanent cure. So, which impacts the brain, which is because of the degeneration of nervous tissue. So, nervous tissue which have got degenerated cannot be generated after a certain age, especially in the old age, they cannot be regenerated. So, stem cell technology is promising you to regenerate neurons and reduce these diseases known as Parkinson's kind of disease, which are considered to be disease which cannot be cured forever. But this is a technology which is promising you to cure in the upcoming future, which leads to a disorder because of degeneration of neurons. So, after knowing what stem cells are, stem cells have the regenerating capability. The meaning of the word cloning is creating the exact copy of or mirror copy of. So, selecting certain stem cells from your body or from an animal, we can clone, replenish and differentiate to create a complete organism itself. So, the phenomena of creating an organism completely means a complete animal or a human being completely with the help of certain cells being derived from the developing embryo, or sometimes the secondary or other perinatal stem cells. When you have got the capacity to make a complete organism is known as cloning. Cloning means exactly the mirror image or exactly the replica of is known as cloning. Cloning technology allows us to generate a population of genetically identical molecules, cells, plants or animals. So, in this process of cloning, we can not only manufacture the complete organism, an organ can be created, a cell can be created or molecules can be created as per our requirement in different conditions. So, in this process, how this process of cloning is done? These are the two techniques which are being used in the process of cloning. These two techniques are being described as artificial embryo twinning is one technology. The second technology is somatic cell nuclear transfer. These are the two technologies which are being used for the purpose of cloning. As you know, the first known animal which is being cloned is a dolly, a sheep. So, in the case of artificial embryo twinning, which is being done by copying nature to the process of twins in nature. So, in the case of twins, as we have got two different variant types of twins, one is monozygotic twins, the other is dizygotic twins. Monozygotic twins can be considered as a natural clones, but dizygotic twins are equivalent to siblings, because in the case of dizygotic twins, 2 eggs are being generated in the mother in a month, and these 2 eggs are being fertilized by 2 different sperms, because of which genetically they are equivalent to siblings. But in the case of monozygotic twins are the resultant of mitosis cell division of the zygote. So, when the zygote, which is the resultant of the fusion of sperm and ova, when it after fertilization, because of mitosis cell division, which divides into 2 cells, in the mitosis cell division, the daughter cells also have the same number of chromosomes. After this division, when the 2 divided cells independently develop into an organism, naturally leads to monozygotic twins, and they are genetically identical, because the complete genome in both the cells is the same. They look alike as it is. That happens in the case of monozygotic twins. So, artificial embryo twinning is a deliberate division of a zygote into 2. In this process, both the individuals develop identically. So, this is known as artificial embryo twinning. So, cloning is development of an exact replica of. So, because they have got the same genome because of the mitosis cell division, and they develop into separate individuals genetically identical, which is known as artificial embryo twinning. So, here in this case of artificial embryo twinning, see, this is a zygote. So, what happens? This morula or a zygote can be divided into 2 equal halves, twinning of zygote. Each part of the zygote, which is the resultant of the division of the morula, will be able to develop into 2 different individuals. So, because they have got the same genomic sequence, the individuals also resemble the same, because they have been derived from a single egg. In the case of dizygotic twins, the mother generates 2 eggs in a month. In this case, you have got a separate egg, because of which the twins are different. In this case of twins are considered here, when you relatively compare with monozygotic twins and dizygotic twins, when the sexes are compared, in the monozygotic twins, the possibility of sexes are both the children will be male or female in the case of monozygotic twins. In the case of dizygotic twins, the sexes possibilities are, one is, in one case the probability is both of them can be males. The second probability is both of them can be females. The third possibility is one can be male and the third can be female. This is the possibility in the case of dizygotic twins, but in the case of monozygotic twins, both of them will be male or female because they have been derived from a single egg or zygote. So, this is known as artificial embryo twinning, and now you are able to differentiate between monozygotic twins and dizygotic twins, and now you have come to know the probability of sexes in the case of monozygotic twins and dizygotic twins. In the case of monozygotic twins, the sex will be either both of them will be males or females, but in the case of dizygotic twin, they can be male, both of them, they can be females or a male or a female. So, the second type of cloning is a somatic cell nuclear transfer. So, here in this case of somatic cell nuclear transfer is considered, how this technology is done. So, for this we need to understand the meaning is in the name of the technology itself, somatic cell nuclear transfer. So, what are somatic cells? We need to know. When you know what somatic cell is, you will be able to understand what somatic cell nuclear transfer is. So, in the case of somatic cells are considered, what happens is our body completely is made up of somatic cells except the germ cells known as sperm and ova. The rest of the body is made up of somatic cells. So, somatic cell nuclear transfer means you are selecting certain somatic cells like subcutaneous cells or some cells which have got some differentiation capability. Each cell has got certain nucleus here. See this, you have got a cell and you have got a nucleus. So, you have removed the nucleus of the somatic cell, means any cell in your body contains cytoplasm and nucleus. The nucleus contains the complete genome. The nucleus contains the complete genome. Genome is the complete set of chromosomes essential for the manifestation of an organism. The physical manifestation is coded in terms of genes in the nucleus in the form of DNA. A sequence of DNA molecule together form genes, genes together form chromosomes, chromosomes together form the complete information of an organism known as genome, is present in every somatic cell nucleus. To understand this technology, this is what we need to be clear with. So, there is a complete information embedded, coded inside the nucleus of every somatic cell in the form of a DNA sequence forming a complete genome. So, here the question is why, which is in a diploid condition. Diploid condition means having a complete number of chromosomes required for the formation of an organism, or having the complete set of chromosomes derived from the mother and father, known as a diploid condition. In the human beings, the diploid condition is having 46 number of chromosomes, which is essential for the generation of an organism. So, here in this case what we are doing is we have removed the nucleus of the somatic cell and we have taken an egg, removed the nucleus of the egg. So, nucleus of the egg, and in the place of the nucleus of the egg we have placed the nucleus of a somatic cell, and the cell assumes that it has been fertilized because the chromosomal condition is complete in a somatic cell which contains 46 number of chromosomes, where an unfertilized egg contains only 23 chromosomes, which will not be sufficient enough to generate a complete organism. So, in this context, egg assumes that it has got fertilized and it develops into a complete new organism, and this resembles exactly the human being from whom we have collected the tissue from the donor. So, this is how cloning is being done. These are the two technologies. One is artificial embryo tuning and the second is somatic cell nuclear transfer. So, thank you friends. We shall meet in the next session discussing other topics in science and technology. Thank you.